Introduction: A hidden thread through time. In the summer of 2017, an international team of geneticists published findings that would reshape our understanding of human history in the South Caucasus region. Their discovery, presented in the prestigious journal Current Biology, revealed something extraordinary. The maternal genetic lineage of people living in Armenia today is virtually identical to those who inhabited the same territory nearly 8,000 years ago. This revelation challenges everything we thought we knew about population movements, cultural transformations, and the genetic roots of civilizations. It tells a story of remarkable continuity, an unbroken maternal thread connecting modern Armenians to their ancestors who lived in the Neolithic period, through the Bronze and Iron Ages, and across millennia of tumultuous historical changes. This discovery is not merely an academic curiosity. It represents one of the longest documented instances of genetic continuity in any human population, providing a unique window into how civilizations can experience dramatic cultural transformations while maintaining genetic stability. For Armenians, it offers scientific validation of deep-rooted connections to their homeland, a genetic passport stamped with the authority of ancient DNA analysis. For anthropologists and historians, it raises profound questions about how cultural change occurs, what drives human migration, and how populations maintain identity across centuries of upheaval. The research breakthrough, Unlocking Ancient DNA, scholars analyzed the complete mitochondrial genomes of 52 ancient skeletons from present-day Armenia and Artsakh, spanning 7,800 years, and combined this dataset with 206 mitochondrial genomes of modern Armenians. This ambitious research project, led by Ashat Margarian and Morten E. Allentoft from the Center for Geogenetics at the Natural History Museum of Denmark, represented a landmark achievement in ancient DNA sequencing. The methodology was rigorous and scientifically sophisticated. Researchers extracted DNA from skeletal remains carefully excavated from archaeological sites across Armenia and Artsakh, representing multiple historical periods. The ancient skeletal samples came from 19 archaeological sites, representing different cultural periods spanning from approximately 300 to 7,800 years ago, including distinct cultural transitions: Neolithic, Chalcolithic, Kura Araxes, Trialeti Vanadzor II, Lichashan Metzamor, Urartian, and Armenian. Classical medieval periods. The technical requirements were extraordinary. Ancient DNA is fragmented, degraded by millennia of burial, and vulnerable to contamination. Researchers had to employ specialized protocols to extract, amplify, and sequence these precious genetic fragments while maintaining strict contamination controls. The CT transition rates at the first position of sequenced DNA fragments were between 8.9% to 43.7%, indicating that the profiled DNA molecules were of ancient origin, with estimated DNA contamination levels under 8%, averaging 1.3% across the entire ancient dataset. The comparison group was equally comprehensive. The ancient mitochondrial genome data was combined with 206 mitochondrial genomes of modern Armenians and previously published data from 7 neighboring populations. This provided context, allowing researchers to determine whether Armenians were genetically unique or whether their pattern of continuity was shared with neighboring populations. Understanding mitochondrial DNA, following the maternal line. To appreciate why this discovery is so significant, one must understand what mitochondrial DNA represents and why, unlike nuclear DNA, which is inherited from both parents, mitochondrial DNA is inherited almost exclusively through the maternal line, passed from mother to child with minimal paternal contribution. This unique inheritance pattern makes mitochondrial DNA an ideal tool for tracing maternal lineages through time. Every woman passes her mitochondrial DNA to all her children. Men pass their mitochondrial DNA to none of their children. Over generations, mutations accumulate in mitochondrial DNA at a relatively predictable rate. Creating a mitochondrial DNA with modern sequences, researchers can reconstruct the maternal genetic history of populations. The study of mitochondrial genomes enables scientists to trace the unique history of females over time, and the ancient mitochondrial DNA evidence finds no evidence of any upheaval over the last 8,000 years. This stability is remarkable, given the turbulent history of the Armenian region. The central finding: 8,000 years of genetic continuity. The primary finding was startling in its clarity. Researchers found genetic continuity for at least 8,000 years in parts of the South Caucasus, with no detectable changes to the female gene pool over this very long time frame. To contextualize this chronological span, 8,000 years ago, the pyramids of Egypt had not yet been constructed. Ancient civilizations in Mesopotamia were just beginning to develop writing systems, and the Americas were inhabited by hunter-gatherer societies. The results clearly indicate genetic continuity of human maternal gene pool since Neolithic times, despite well-documented cultural shifts in the South Caucasus. This represents perhaps one of the most striking paradoxes in human genetic history. A region documented by archaeologists to have experienced profound cultural transformations maintained genetic stability in its maternal population. The genetic analyses employed multiple sophisticated methods to test whether this apparent continuity was statistically real or merely coincidence. Researchers tested 5 different demographic scenarios explaining the formation of the modern Armenian gene pool. And despite well-documented cultural shifts across the time period, the results strongly favor genetic continuity in the maternal gene pool. Index SARS 30, here's for 1. The South Caucasus, situated between the Black and Caspian Seas, geographically links Europe with the Near East, and has served as a crossroad for human replacement as different groups migrated through or invaded the region. Yet the genetic evidence suggests otherwise. The mountainous terrain of Armenia may have played a crucial role in this protection. High mountains create natural barriers to movement, potentially insulating local populations from external genetic influx. The Armenian highlands, with elevations often exceeding 1,500 meters, form a distinct geographical unit. This isolation, combined with other factors, may have preserved the maternal gene pool even as foreign armies marched through the region and invaders came and went. The mountainous landscape, a distinct and very old Armenian language, and a strong national and cultural identity, reinforced later by the adoption of Christianity, might have been conducive for the long-term genetic isolation of Armenians from neighboring populations. Geography, language, culture, and religion all conspired to maintain genetic continuity even when political control changed hands repeatedly. The paradox. Cultural transformation without genetic revolution. One of the most intellectually intriguing aspects of this discovery is the disconnect between documented cultural change and genetic stability. Over the 8,000-year period examined, Armenia experienced numerous distinct cultural periods, each associated with different archaeological signatures, different settlement patterns, and different material cultures. Kura Araxes culture, characterized by distinctive pottery and burial practices. Later came Urartian civilization, a complex state, with cities, written records, and sophisticated administration. Then came Greek influence following Alexander's conquests, Roman dominance, and eventually the transformation of Armenia into an early Christian nation in 301 CE. Yet through all these transformations, the maternal genetic pool remained stable. Findings suggest either that cultural shifts occurred primarily through the exchange of ideas, or that it was primarily men who moved into new territories, bringing new cultural ideas along with them. This interpretation is profound. Ideas can travel rapidly. Genetic material travels only when people move. If invading armies were predominantly male, they might introduce new ideas, religions, and cultural practices without substantially altering the local maternal gene pool. The complete picture. Admixture events and the modern gene pool. While the maternal genetic continuity is remarkable, the complete genetic picture of modern Armenians is more complex. Using genetic data of modern Armenians, researchers suggest that the Armenian gene pool was formed as a result of admixture events happening approximately 4,500 years ago. This indicates that while the maternal lineage remained continuous, paternal ancestry and overall genetic composition changed significantly. This pattern, stability in maternal lineage combined with admixture in the overall gene pool, suggests that male-mediated gene flow was substantial, but female gene flow remained limited. Invading or migrating populations were predominantly male, introducing new patrilineal ancestry without substantially altering the maternal genetic composition. Over millennia, this created a modern Armenian population with complex ancestry, continuous maternal lines stretching back 8,000 years, combined with more recent paternal ancestry from admixture events. Recent comprehensive genome-wide studies have expanded this picture further. The first whole genome study challenges the long-held belief based on Herodotus's theory regarding Armenian origins from the Balkans, revealing no significant genetic link between Armenians and the populations in the Balkan region. Instead, Armenian ancestry appears rooted in the Caucasus and broader Near East, not in European Balkans as classical sources suggested, the living fossils. Concept: Armenians as genetic continuity. Some scholars have provocatively referred to modern Armenians as living fossils, not in a derogatory sense, but to highlight the extraordinary genetic continuity they represent. Several scholars referred to modern Armenians as living fossils, due to these discoveries demonstrating genetic continuity extending back beyond the Bronze Age to 7,111 years ago. This characterization, while requiring careful interpretation, reflects the uniqueness of Armenian genetic stability. Most human populations have experienced more substantial genetic turnover over similar time periods, with migrations, invasion, and admixture events reshaping genetic composition. Armenia's relative isolation, combined with cultural and linguistic distinctiveness, preserved a genetic continuity that is rare, perhaps even unique in human history. The implication is profound for Armenian identity. Armenians are not merely carriers of a distant cultural and linguistic tradition, they are biological descendants of the Neolithic inhabitants of their region. The connection to the land and the historical Armenian highlands is not metaphorical but literal, written in the DNA of the female lineage in each modern Armenian. More recent research has added nuance to this picture by examining geographic variation within Armenia. Researchers found differences in the timing of Levantine-related ancestry's penetration into various parts of the highlands, with the western parts receiving this ancestry during the Chalcolithic period, while the eastern parts received it through admixture occurring sometime after the early Bronze Age. This suggests that genetic continuity was not uniform across Armenia. The eastern regions maintained stronger continuity with Neolithic populations, while western regions experienced more substantial foreign genetic input during the Chalcolithic period, possibly from populations linked to Neolithic Levantine farmers. This geographic variation likely reflects the differential vulnerability of different regions to invasion and population movement. Some areas were more isolated and protected, while others were more exposed to foreign influence. The revelation indicates a high level of regional genetic continuity in eastern parts of Armenia across an 8,000-year span. Despite the region's experience of multiple cultural transitions, eastern Armenia's greater isolation may have preserved maternal genetic continuity even more completely than western regions, which experienced more substantial admixture. The broader implications: rewriting population history. This discovery has significant implications for our understanding of how populations maintain identity and how genetic and cultural changes occur. It demonstrates that populations need not experience complete genetic replacement to undergo dramatic cultural transformations. Ideas, religions, and technologies can spread without necessarily displacing resident populations. It also challenges narratives that attribute all cultural change to invasion and population replacement. While male-mediated gene flow did occur in Armenia, as evidenced by admixture signals in the nuclear genome, this genetic influx did not completely replace the existing population. Instead, foreign males integrated into local populations, contributing new genetic material, while the established female population continued its unbroken lineage. For archaeologists and historians, this suggests a model of cultural change driven partly by idea transmission, partly by elite replacement and cultural prestige, and partly by limited male-mediated gene flow, but not by complete population replacement. This nuanced model better explains the archaeological evidence of cultural transformation alongside genetic continuity. Recent updates. The 2024 whole genome study. While the 2017 discovery of 8,000-year maternal genetic continuity remains fundamental, recent research has added additional layers of understanding. In 2024, researchers published the first comprehensive whole genome study of ancient and modern Armenian populations, revealing additional insights about Armenian demographic history. The 2024 whole genome study challenges the long-held belief, based on classical historian Herodotus' theory of Armenian origins in the Balkans, revealing no significant genetic link between Armenians and Balkan populations. Instead, Armenian ancestry appears to derive from the South Caucasus and Near East regions, with complex admixture from multiple sources over time. The research documented genetic input into the region from sources linked to Neolithic Levantine farmers at different time periods in different regions, during the Chalcolithic in western Armenia, and after the early Bronze Age in eastern Armenia. This refined understanding suggests that Armenian genetic history reflects a complex interplay of local population continuity and external gene flow, with geographic variation reflecting different regional histories. The ancient DNA revolution. Technology enabling discovery. The ability to make these discoveries reflects revolutionary advances in ancient DNA technology. When the field of ancient DNA began in the 1980s and 1990s, researchers could extract only small DNA fragments from carefully preserved samples. Modern technology permits extraction and sequencing of complete mitochondrial genomes, all 16,569 base pairs, from degraded ancient samples. These technological advances have transformed population genetics, allowing researchers to literally read the genetic history written in ancient bones. The Armenian research was possible only because researchers could extract DNA from skeletal samples using protocols that minimize contamination, amplify specific DNA regions using polymerase chain reaction, PCR, sequence complete mitochondrial genomes to high depth, compare ancient sequences with modern populations using sophisticated phylogenetic methods. Each of these capabilities represents decades of methodological development and refinement. The Armenian study represents one culmination of this decades-long scientific journey. Cultural significance, genetic evidence of home.